Every second, millions of cells in your body divide. But before a single cell can divide, it must complete one extraordinary task. When a cell divides, both daughter cells need this complete genetic blueprint. DNA achieves this through a process called semiconservative replication. The process begins with Helicase. This molecular machine moves along the DNA, breaking the hydrogen bonds between the two strands. Each new DNA molecule keeps one original strand and builds one completely new complementary strand. The original strand serves as a template. Helicase unzips the double helix, exposing the genetic code on each strand. But DNA polymerase cannot start building on its own. So Primase first places short RNA primers on both strands, giving DNA polymerase a starting point to begin synthesis. Now DNA polymerase begins copying the DNA. It follows one simple rule: adenine pairs with thymine and guanine pairs with cytosine. There is one important limitation. DNA polymerase can only build a new strand in the 5' to 3' direction. This single rule creates two very different types of strands at the replication fork, each copied by a different mechanism, creating an almost perfect copy of its entire DNA. DNA is the blueprint of life, storing every instruction needed to build and maintain your body. The leading strand is copied continuously because DNA polymerase can move in the same direction as the replication fork opens. The opposite strand, called the lagging strand, cannot be copied continuously. Instead, it is built in short sections called Okazaki Fragments. Each Okazaki fragment starts with a new RNA primer laid down by Primase, and DNA polymerase extends it in the 5' to 3' direction. Finally, DNA Ligase joins all the Okazaki fragments together, sealing the gaps to produce one continuous DNA strand. The result is two identical DNA molecules. Each contains one original strand and one newly synthesized strand. This is exactly as described by semiconservative replication. The original genetic information is perfectly conserved in both copies. Remember the sequence for NEET. Helicase unzips the DNA, Primase lays the primer, DNA polymerase builds the new strand, and Ligase seals the fragments. Master these 4 enzymes and DNA replication becomes one of the easiest chapters in biology.